The topic covered in this video is illumina sequencing, which is basically a method of figuring out the sequence of DNA segments. So first of all, what is DNA? DNA is a macromolecule that is a chain of nucleic acids, and our nucleic acids are adenine, thymine, guanine, and cytosine. And we're interested in these because the arrangement of these nucleotides gives cells instructions in building RNAs and therefore protein. So the reasons we study sequencing, or the reasons we sequence, are because we're getting the blueprint of the cell by getting the order of these nucleotides. We can look at specific genes, we can look at regulatory regions and variances in these genes between individuals. We can look into diseases like cancer, psychiatric diseases, autoimmune ones that may relate to the genes that somebody has. We can also study microbes like unculturable organisms, viruses that don't grow on their own, and host pathogen interactions. And then we can apply this science to things like agriculture when we're looking at cultivation or making breeding decisions. So there are 4 steps in illumina sequencing. There's sample prep, cluster generation, sequencing, and data analysis. And I'll go over all of these steps, which are explained in another video made by the makers of illumina that goes much faster than this video. So first of all, sample prep. We have to take our large DNA segments and we break them into small fragments, which will be represented by this chain of circles here. So once we have our sample broken up into fragments, we add adapters onto each side, seen here. So these adapters contain sequencing binding sites, indices, and oligo binding sites, which I'll talk about in just a second. So once we have our sample prepped, we go on to cluster generation. So the first step is adding our sample to this flow cell, which is basically just a glass slide that has these little oligos here bound down to it. So you'll notice that these oligos look very similar to our adapters that were added in the last step. And that's because they act as a reverse complement, so that when our sample is added, it binds to those oligos. So once we have our sample bound, we add in polymerase, which comes in and makes a complementary strand to our original sample. And then we can wash the original sample off, and now we have a reverse complement of our sample that is tethered to this flow cell and can't be removed. So now that we have this reverse complement, we actually want an exact copy of our strand of DNA in order to do our first set of sampling. So we form this bridge between the reverse complement of our sample and the second type of oligo that's found on this flow cell. We add in polymerase again, and it comes in and adds on, let me do that slower. So it comes in and adds on nucleotides again, and it is the reverse complement of our reverse complement. And so now we have 2 copies of this gene. One is the reverse complement and one is an exact copy seen here. So we denature the bridge to make our sample stand up like this. We repeat this process a few thousand times, and it makes thousands of copies of both of these 2 samples here. And then everything that represents the reverse complement of our original sample is washed off. And then 3' ends are bound, which would be here and here. And this makes sure that when we're doing our sampling, the nucleotides are added in this direction and not here at the end of our sample. So then we move on to sequencing. So the first step of sequencing is binding a primer to the adapter, which you'll see right now. And then one at a time, nucleotides are added on to this primer. But you'll notice that these nucleotides have a little something extra added onto them that these don't, and that's this little fluorescent tag. So when this nucleotide is added, a light is shown onto this sample, and it will shine a light back at the machine, which can be read by the machine. So each color represents a different nucleotide. In this example, blue is A, red is T, green is G, and yellow is C. So far our first nucleotide was an A, The second one shined red, so it was a T. The third one shined green, so it was a G. Yellow is C. And then blue for another A. So we know that this sample right here was A, T, G, C, A. And since each nucleotide only binds to a specific other nucleotide, we therefore know that this section right here has a sequence of T, A, C, G, T. So as you notice, this sequence only goes about halfway down this sample right here. So in order to get the rest of our sample, we need to wash the product that we made off, and then we form a bridge going in the opposite direction. So this is the reverse complement that we made the first time we copied our DNA strand. So then we basically do the exact same process. We add this adapter here, and we start adding on nucleotides one at a time, watching for those light reactions. And it basically, this time, the sequence in this direction of this piece of DNA can be directly recorded, and we know the sample of our original sample. This right here shows a picture of what an illumina plate looks like when one burst of light is sent out. And so basically it can take these individual lights and they'll change each time a signal or each time a nucleotide is added on. So all of these lights represent a different piece of the DNA that we originally put in. And then it'll take these pictures and it'll stack the lights on top of each other. So this is kind of a bad example because it makes it look like every piece of DNA has a sequence of T, G, C, A, but it gives you an idea of what's going on. So then we move on to the analysis step. So reads with similar sequences are clustered together and they're basically assumed to be the same piece of DNA, or they're considered to be pieces of neighboring DNA. And so what you're seeing here is two pieces of DNA that to us look like a jumbled mess, but when they're aligned properly, you see that this part right here is exactly the same in both of these sequences. And so it's assumed that these two pieces of DNA came from a similar place, just on a different, or from the same chromosome in different cells. And so we should also expect that if this piece of DNA were continued in this direction, then we know that the sequence before it is A, G, T, T, C. We have this sequence, and then now we have what goes on the other end as well. So this whole piece right here comes together to make one long strand of DNA. And then we take overlapping sequences and use it to reconstruct the original unbroken strand. So originally we were looking at pieces of DNA that had large chunks that overlapped. And now we begin looking at pieces of DNA that had smaller overlaps. So the pieces here, red and purple, were read in the reverse reading direction, so the first read that we got. And then the ones over here in blue were taken from the reverse direction, so the second read. And so in order to read these pieces together, we need to get a reverse complement of these blue strands right here and read them in the direction that they would be read if they were read first, which is seen now in purple. So we start looking for overlapping regions, and we see that this region here and this region here overlap at this TACG site. So we can assume that in this sample, this piece comes before this, the red parts are the same, and then this part comes afterwards. Same thing here with the yellows. This sequence comes first, this is in the middle of the two sequences, and then this part right here comes afterwards. And again with the green, exact same thing. And so then we can take these four pieces of DNA and we can line them up based on the way that these little short overlapping segments appear. And we can make one long strand of DNA. and we can start to piece together an entire genome like this. This is a link to the website that I did not plagiarize when I was making this video. Really quickly, what we can do with this is we can sequence entire genomes in 24 hours, which is relatively fast considering how much DNA there is in a single cell. We can look at variations within DNA and apply this to cancer patients or other individuals with genetic disorders. We can look for favorable traits in livestock and compare bacterial genomes to each other.